NanoCas helps to unlock Mini-CRISPR. First march toward miniature gene editing. Genetic disorders, characterized by mutations that alter the genetic material over generations, can be mendelian or chromosomal. The former is identified by an alteration in a single gene and encompasses diseases like hemophilia, thalassemia, cystic fibrosis, Duchenne muscular dystrophy, and sickle cell anemia. The alteration is traced by performing a pedigree analysis across generations, for by the word of Mendel, dominant and recessive traits capture inheritance at length. Chromosomal disorders, on the other hand, are identified by an absence, excess, or change in the structure of chromosomes and include Down syndrome, Turner syndrome, Klinefelter syndrome, and Williams syndrome. One of the solutions to genetic disorders is to reverse mutations by deleting, inserting, replacing, or modifying DNA. On the surface, gene editing can seem like a piece of cake. After all, it's a code, a sequence of molecules, and all one needs to do is alter that sequence. What could possibly go wrong? It turns out, a small mistake in gene editing can cause major problems. At its core, genetic editing deals with rewriting the genetic instructions. You could unintentionally target the wrong DNA sequences, introducing a new genetic mutation that could turn fatal. Even if you get the sequence right, you then have to correctly deliver the editing machinery. A correct delivery of the editing machinery might still face challenges from the immune response, and the cycle that follows suit would be a long battle between the genetic makeup and the immune system, two crucial stakeholders that decide the health of a person. Gene editing strongly relies on precision, accuracy, efficient delivery of the editing machinery, and controlled editing of the output. Keeping in mind these restraints, Professor Jennifer Doudna of UC Berkeley and Professor Emmanuel Charpentier of Max Planck Unit for the Science of Pathogens revealed that the CRISPR-Cas9 bacterial immune system could be repurposed as a gene editing tool, subsequently winning the Nobel Prize in 2020 for their groundbreaking research. An acronym for Clustered Regularly Interspaced Short Palindromic Repeats, CRISPR. Cas9 is a component of the bacterial immune system that scientists adapted to use like a set of molecular scissors to precisely cut DNA, enabling it to be a repurposed gene editing tool. First discovered in bacterial immune systems by Francisco Mojica, CRISPR was found to cut the DNA of bacteriophages, viruses that invade bacteria, and disable them. CRISPR consists of two key components, a CRISP-associated, Cas nuclease, that binds and cuts DNA, and a guide RNA sequence, GRNA, that guides the Cas nuclease to its target. By modifying Cas9, researchers have been able to control gene editing with focused ultrasound, FUS, turning on certain genes and study them for disease research. This process allows researchers to target and modify different errors that occur in the massive sequence of the human genome, thus marking the first step towards treating genetic disease. Despite its reputation as an efficient and customizable alternative to other gene editing tools, and U.S. regulator approval to perform gene editing in sickle cell disease in 2023, the in vivo delivery of CRISPR using the adeno-associated virus, AAV, a virus commonly used as a ferry for CRISPR, has been described by researchers as complicated. The main difficulty is the bulky nature of the components of CRISPR, particularly the 1,300 amino acid Cas9 enzyme employed to cut the DNA. This amount of amino acids translates to proteins with a weight of 160 kilodaltons, 320 kilograms, which is too much to stuff into a 25 nanometer AAV. Researchers tried delivering the CRISPR and the guide RNA through two AAVs, but developments showed no success. Recently, Mammoth Biosciences, the company co-founded by UC Berkeley's Nobel Prize-winning scientist, Professor Doudna, rigorously screened the genetic sequence data of 176 CRISPR varieties found in microbes, picking NanoCas as the miniature successor to CRISPR. The chief scientific officer and co-founder of the company, Lucas Harrison, and his colleagues have optimized NanoCas by modifying its proteins. Their research is presented in a paper in Bio-Ziv, titled Single-AAV CRISPR Editing of Skeletal Muscle in Non-Human Primates with NanoCas, an ultra-compact nuclease. The paper presents NanoCas as having potent editing capabilities equaling that of first-generation CRISPR systems. NanoCas is a much smaller version of CRISPR, with only 425 amino acids, about one-third the size. Its small size makes it easier to squeeze into an AAV. In tests on mice, the team at Mammoth Biosciences found that NanoCas successfully edited the PCSK9 gene, a protein that regulates cholesterol levels, in the liver with about 60% efficiency, performing as well as SACAS9, a much bigger CRISPR-based tool. To add to this ability of NanoCas, Researchers also found that, like SACUS9, NanoCas reduced the PCSK9 protein levels to undetectable limits, which could have important implications for treating and preventing cardiovascular diseases. Further, in a mouse that exhibited Duchenne muscular dystrophy, a muscle wasting disease that arises from a mutation in the dystrophin gene, NanoCas was able to insert genetic material in about 10-40% of tissue, specifically in the quadricep, calf, and heart muscles when inserted through a single vector AAV. Finally, for the first time ever, researchers performed single AAV muscle editing in 3 healthy macaque monkeys. They injected an AAV, which contained the NanoCas, into the monkey's bloodstream. NanoCas achieved an efficiency of about 30% when focusing on dystrophin mutations in the skeletal muscles of the macaque species, with a 15% editing efficiency in the heart, a better accuracy than its parent CRISPR with minimum off-target editing. Trevor Martin, the CEO of Mammoth Biosciences, attributed NanoCas achievements to its compact size and compatibility with a wide range of gene editing techniques. He also added that from facing potential problems in editing tissues outside the liver to making a major step towards miniature editing has been remarkable. Terence Flott, the dean of University of Massachusetts T.H. Chan School of Medicine and gene therapist, hailed these levels as clinically meaningful and, despite expressing concerns about off-target editing, remained hopeful due to its ease of delivery via AAV and compatibility. Harrington seconds these challenges and has promised that he and his team at Mammoth Biosciences would work on these challenges by allowing it to target other more inaccessible regions relevant to genetic diseases through the AAV. Mammoth's belief in the therapeutic capabilities of CRISPR and their unwavering dedication and devotion, driven by the goal to make treatments accessible for patients with genetic disorders to improve their lives, is what has steered them towards presenting this wonderful innovation to the world. Their work may one day make the dream of accurate and precise gene editing through NanoCas a reality.
Single-AAV CRISPR editing of skeletal muscle in non-human primates with NanoCas, an ultra-compact nuclease bench. CRISPR gene editing is a transformative technology for addressing genetic diseases, but delivery constraints have largely limited its therapeutic applications to liver-targeted and ex vivo therapies. Here, we present the discovery and engineering of NanoCas, an ultra-compact CRISPR nuclease capable of extending CRISPR's reach in vivo beyond liver targets. We experimentally screened 176 ultra-compact CRISPR systems found in metagenomic data and applied protein engineering approaches to enhance the editing efficiency of NanoCas. The optimized NanoCas exhibits potent editing capabilities across various cell systems and tissues in vivo when administered via adeno-associated viral AAV vectors. This is accomplished despite NanoCas being approximately one-third the size of conventional CRISPR nucleases. In proof-of-concept experiments, we observed robust editing with our optimized NanoCas in mouse models targeting PCSK9, a gene involved in cholesterol regulation, and targeting exon splice sites in dystrophin to address Duchenne muscular dystrophy, DMD mutations. We further tested the efficacy of our NanoCas system in vivo in non-human primates, NHPs, resulting in editing levels above 30% in muscle tissues. The compact size of NanoCas, in combination with robust nuclease editing, opens the door for single AAV editing of non-liver tissues in vivo, including the use of newer editing modalities such as reverse transcriptase, RT editing, base editing, and epigenetic editing. Introduction: CRISPR-Cas systems have revolutionized the field of genetics, offering unprecedented precision in gene editing and presenting viable strategies for correcting genetic diseases. Due to their large size, existing CRISPR-Cas systems, including Cas9, 1,368 amino acids, and Cas12a, 1,307 amino acids, while effective, exceed the packaging capacity of single adeno-associated viruses, AAVs, the most widely used vectors for gene delivery in vivo. 1. Figure 1a, extended data. Figure 1b. As a result, CRISPR gene editing's clinical applications have been 1. Constrained to ex vivo therapies and liver-directed therapies where lipid nanoparticles, LNPs, can be used. A compact, potent CRISPR system able to comfortably fit within the AAV genome would overcome these constraints, enabling specific in vivo gene editing of diverse tissues including the central nervous system, skeletal and cardiac muscle, which are challenging to edit by other methods too. After the establishment of Cas9 in 2012, extensive efforts have focused on exploring microbial CRISPR diversity and characterizing alternative genome editing platforms. Figure 1b, 3-7. Recent analyses suggest that natural diversity encompasses hundreds of thousands of CRISPR systems. 8. The identification of smaller Cas proteins, such as Cas12I, and say Cas9, has partially addressed the size limitations inherent to Cas96, 9, 10. However, even these compact variants remain too large for efficient AAV delivery when combined with necessary regulatory elements and fusion proteins. Figure 1a. While dual AAV delivery approaches have been attempted to overcome size limitations, these strategies suffer from significantly reduced editing efficiency and create additional manufacturing and safety challenges due to the need to deliver multiple vectors. This size constraint particularly impacts newer editing modalities that rely on fused effector proteins to edit DNA, including reverse transcriptase editing 11, base editing 12, and epigenetic editing 13. The smallest known CRISPR systems to date include Cas14, Cas12F, and ancestral TNPB, and ISCR proteins 14-19. While promising, these systems demonstrate lower editing efficiency compared to larger systems. Their therapeutic potential remains largely theoretical, with limited in vivo studies and no published demonstrations of their efficacy in large animal models such as non-human primates, NHPs. Successful development of new gene editing therapeutics requires robust validation in NHPs, as their high genetic homology with humans and similar tissue architecture and immune responses make them the gold standard for predicting clinical translatability. 20. Furthermore, these systems face additional challenges, including complex protaspacer adjacent motif, PAM sequences, inconsistent guide RNA performance, and lengthy guide RNA sequences, which restrict their utility in therapeutic applications. These limitations in current CRISPR technology led us to hypothesize that the vast diversity of natural CRISPR systems, combined with advances in protein and RNA engineering, could yield an ultra-compact CRISPR system capable of efficient editing within a single AAV vector. We developed a comprehensive screening strategy that simultaneously evaluated multiple parameters critical for therapeutic editing, including size, editing efficiency, specificity, and compatibility with diverse target sequences. Extended data. Figure 1a. Through this approach, we conducted one of the most extensive screenings of small CRISPR systems to date, synthesizing and experimentally analyzing 176 natural candidate editors to identify and engineer our leading candidate, NanoCas. NanoCas, with a protein size of approximately 450 amino acids, is roughly one-third the length of Cas9 and Cas12 and half the size of previously described compact systems like Cas12I, Cascs, or say Cas9. Figure 1a. Extended data. Figure 1b. Through our nuclease discovery and 2 engineering efforts, we have demonstrated that NanoCas achieves robust editing efficiency despite its small size. Here we establish NanoCas as an effective tool for in vivo genome editing, including through successful editing in non-human primates using a single AAV that targets muscle cells. This work expands the therapeutic possibilities of in vivo CRISPR beyond liver-targeted applications, enabling efficient gene editing of previously difficult-to-reach tissues, and ultimately opens a route to treatment of a number of genetic disorders not addressable with current technologies. Results. Discovery of NanoCas. Among a cohort of ultra-compact CRISPR systems, we began our search for novel CRISPR systems by establishing a target editor profile, TEP, that defined key parameters for therapeutic applications. Extended data. Figure 1a. The ideal system would maintain Cas9 equivalent editing efficiency, be smaller than 600 amino acids, target 10% of genomic sequences through PAM recognition, achieve successful editing with 30% of designed GRNAs, match Cas9's fidelity, and use an RNA component under 100 nucleotides. Using this TEP as our guide, we conducted a systematic search of 21,980 metagenomes using hidden Markov models, HUMs, built from known compact type V CRISPR systems. This initial screen identified 6,945 candidates under 600 amino acids, of which 3,958 contained at least one CRISPR array within 2 kilobases of the nuclease gene. We implemented a three-stage screening strategy to evaluate these candidates. 1. Computational RNA structure prediction. 2. PAM identification and RNA component validation in mammalian cell lysates. And 3. Assessment of chromosomal editing in mammalian cells. Fig. 1c. Our initial focus was on proteins related to Cas14a1. Based on similarities to Cas14a systems, we hypothesized these candidates would require both a CRISPR-associated RNA, crRNA, and transactivating RNA, tracker RNA, for function. Figure 1b. 12. We identified potential tracker RNAs by analyzing conserved non-coding sequences near Cas genes and CRISPR arrays. As expected, our search revealed a diversity of tracker RNA architectures across different candidate clusters. However, in some cases, disparate candidate clusters shared a conserved tracker RNA architecture featuring 2 stem loops and 2 unpaired segments with sequence complementary to nearby CRISPR repeats, all within 50 nucleotides. Figure 1d. Extended data. Figure 1c. This architecture resembled that of Cas14a-like systems but was significantly more compact. We leveraged this finding to conduct RNA-centric database searches, identifying additional candidates that would have been missed through protein-based searches alone. We designed chimeric guide RNAs for all candidates by joining the tracker RNA to target sequences via a 4-nucleotide linker. Figure 1d. Ultimately testing 176 systems, more than half of which employed the 50NT tracker RNA architecture described above. Evaluate PAM preferences and validate GRNA designs. We expressed candidate nucleases in HEC293 T cells rather than using bacterial or in vitro systems. This approach offered the advantage of selecting for variants that express well in mammalian cells, a common translational bottleneck. 21. We combined cell lysates containing expressed nucleases with synthetic GRNAs and a target plasmid library containing randomized PAM sequences. Figure 1e. Using NGS analysis of cleaved plasmid 17, we identified PAM sequences for 76 candidates, 43% of those screened. Figure 1c, revealing approximately 30 distinct PAM preferences. We then developed a chromosomal editing screen in HEC293 T cells to identify nucleases active in human cells and assess their consistency across different target sites. Figure 2f. As many CRISPR systems showing consistent performance across GRNA sequences, limiting their utility, we tested each candidate with up to 24 unique GRNAs targeting various genes to comprehensively evaluate editing capabilities. Of the 65 systems tested, 20 showed detectable activity with at least one GRNA, though most achieved average editing rates below 5%. One candidate, which we named NanoCas, distinguished itself with superior performance, achieving an average editing rate of 20% and successfully editing with 60% of tested GRNAs. This substantially exceeded other candidates, including our Cas14a1 control, which averaged 8% editing efficiency. Initial assessment in cell lysate revealed NanoCas recognized a TNTR PAM sequence, or TNTN, under relaxed conditions. Figure 1e. Next, we further scrutinized the PAM of NanoCas in human cells to determine its identity in a chromosomal editing context as opposed to the plasmid cleavage assay. NanoCas was screened with 79 guides in total, all with non-inter PAMs, and we were surprised to find substantial flexibility at the -4 and -1 positions, with some spacers showing tolerance of N and Y identities at these positions, respectively. Figure 1g. With the implied NNTN PAM flexibility, NanoCas can natively target 48.7% of bases in the human genome, substantially improving targetability relative to previous compact type V systems 17. Like other type V nucleases, NanoCas generates large deletions distal to the PAM site. Extended data. Figure 1d. Sanger sequencing mapped precise cut sites to positions +22, +23 on the target strand and +27, +28, +29 on the non-target strand relative to the PAM. Extended data. Figure 1e. This comprehensive screening process, from bioinformatic prediction through rigorous functional testing, highlights the rarity of finding highly efficient nucleases in nature. Of thousands of candidates meeting size requirements, only NanoCas emerged with both compact size and robust editing capability. Figure 1c. An engineered NanoCas variant with increased potency to enhance NanoCas's genome editing capabilities. We first investigated its biochemical properties to identify potential limitations. Fluorescence polarization assays using a nuclease-deficient variant revealed notably weak binding affinity to target DNA sequences. Figure 2a. Based on this finding, we engineered variants with positively charged arginine substitutions across the protein to improve DNA binding affinity. Screening these variants in HEC293 T cells identified D220R as the most effective mutation. Figure 2b. This enhanced variant demonstrated improved editing efficiency across a range of mRNA doses in HEC293 T cells. Extended data. Figure 2a. Fluorescence polarization assays confirmed the D220R variant's enhanced DNA binding. Figure 2a. Supporting our hypothesis that wild-type NanoCas's editing efficiency was limited by DNA binding affinity. Understand the molecular basis for D220R's improved performance. We generated a structural model of NanoCas D220R bound to target DNA. Figure 2c. The model places R220 near the target's trans-phosphate backbone at the PAM-target interface, suggesting it enhances DNA binding through sequence-independent interactions. This predicted structure aligns with known architectures of Cas12F systems 22-24, showing two NanoCas protomers bound to a single GNA complex. Given D220R's enhanced DNA binding, we investigated its impact on editing specificity. We assessed mismatch tolerance in T cells using a guide RNA targeting the B2M locus. By introducing single mismatches at each position across the guide sequence, we found that both wild-type NanoCas and D220R maintain similar mismatch sensitivity profiles, with positions 8, 9, and positions 18, 20 showing higher tolerance for mismatches. Figure 2d. Extended data. Figs. 2b. For both variants, mismatch tolerance was particularly pronounced near the PAM distal end, where editing rates reached approximately 40 to 50% with D220R. NanoCas editing in primary cells and in vivo mouse editing via single-AAV in the liver. To evaluate NanoCas's therapeutic potential ex vivo, we generated NanoCas mRNA and confirmed the impact of the D220R mutation through dose-response studies in both T cells, TRAC, and CD34+ HSPCs, BCL11A. Figure 3a. The engineered variant demonstrated superior editing across all mRNA doses tested, reaching 90% efficiency in T cells and 80% in HSPCs, compared to wild-type's lower maximal efficiencies of 80% and 50% respectively. This enhanced performance was particularly evident at lower mRNA doses in both cell types. To evaluate NanoCas's therapeutic potential in vivo, we targeted PCSK9, a clinically validated gene target for cholesterol reduction. We first screened 114 guide RNAs targeting PCSK9 exons through plasmid transfection in HEPA-1-6 cells. Extended data. Figure 2c. After identifying the most effective guide RNA, we constructed AAV8 vectors for delivery in wild-type C57BL/6 mice. For Cas9, we based our AAV on previously used promoters, selecting a minimal TBG promoter 25. For NanoCas, its smaller size enabled us to include additional regulatory elements while maintaining a total cargo size of just 4.3 kilobytes, well under the AAV packaging limit. We leveraged this extra capacity to incorporate both a larger liver-specific ApoHot promoter and a woodchuck hepatitis virus post-transcriptional regulatory element, WPRE, which has been shown to enhance mRNA stability. 26. Figure 3b. Following tail vein administration of 2E13 viral genomes kg, we assessed liver editing efficiency after 2 weeks. Amplicon sequencing of liver tissue revealed that the NanoCas D220R variant achieved robust editing rates of 60%, comparable to Cas9, while wild-type NanoCas reached 15% editing. Figure 3c. The 60% editing efficiency suggests successful modification of the majority of hepatocytes. 27. Importantly, both Cas9 and NanoCas D220R treatments reduced serum PCSK9 protein to undetectable levels, while wild-type NanoCas achieved a 40% reduction. Figure 3D. These results demonstrate that our engineered NanoCas variant matches the effectiveness of the larger Cas9 system while maintaining the advantages of a more compact size. NanoCas editing of humanized dystrophin in mice. Following our success with liver editing, we explored NanoCas's potential for therapeutic genome editing in muscle tissue, focusing on dystrophin, which is mutated in Duchenne muscular dystrophy, DMD. Previously described DMD editing strategies include 1. Disrupting splice acceptor sites and 2. Directly disrupting and reframing premature stop codons, and these strategies represent a promising approach for treating DMD by producing truncated but partially functional dystrophin proteins 28. To evaluate NanoCas's capability for exon skipping, we first screened guide RNAs targeting splice acceptor sites in the dystrophin gene using HEC293 T cells. Figure 4a. We identified 2 promising guides targeting exons 44 and 51, respectively. We packaged these guides along with NanoCas into AAV9-4A vectors, a capsid variant optimized for muscle delivery 29, using the muscle-specific CK-AD promoter 30. Figure 4b. Initial testing in human myotubes demonstrated robust editing efficiency, with NanoCas achieving 20 to 40% editing across a range of viral doses, MWA1E4-1E6, while Cas9 editing remained below 15%. Extended data. Figure 4a. We then evaluated these constructs in a humanized DMD mouse model, administering AD13 of the AAV vectors carrying guide 1 and guide 2, respectively. Analysis of muscle tissue 4 weeks post-injection revealed substantial editing, ranging from 10 to 40% across quadriceps, heart, and calf muscles. Figure 4c. We note that Cas9 could not be evaluated in this model due to PAM sequence incompatibility with a single nucleotide polymorphism in the human dystrophin sequence in this mouse model. NanoCas editing in non-human primates via single-AAV. Building on our successful mouse studies, we evaluated NanoCas in NHPs, a critical model for assessing therapeutic gene editing safety and efficacy. We administered our optimized AAV constructs to cynomolgus macaques, N=3, and collected serial muscle biopsies at 4 and 8 weeks, followed by comprehensive tissue analysis at necropsy. Biopsies of vastus lateralis showed progressive editing, with NanoCas achieving 5 to 20% indels at 4 weeks and increasing to 10 to 30% by 8 weeks. Figure 4d. In contrast, Cas9 treated animals showed minimal editing, 2%, in the same tissue. At necropsy, we analyzed multiple segments, proximal, medial, and distal, of each muscle tissue to assess editing distribution. Despite some variability between segments, comprehensive analysis of skeletal muscle tissues revealed editing levels up to 30% with NanoCas, while Cas9 editing remained below 3%. Figure 4d. 4e. Extended data. Figure 4b. Notably, these editing levels are particularly encouraging given that restoration of approximately 10% of normal dystrophin levels is thought to be sufficient for therapeutic benefit in DMD patients. 31. We also examined editing in cardiac tissue, where NanoCas achieved 15% editing across the left ventricle, left atrium, and septum, compared to 10% with Cas9. Figure 4e. Analysis of liver tissue showed minimal off-target editing, with indel levels below 2% across all 4 lobes. Throughout the study, animals maintained normal aspartate transferase levels after an initial, transient elevation that resolved by day 8. Data not shown. To our knowledge, this represents the first demonstration of efficient muscle editing in NHPs using a single AAV CRISPR system. Previous attempts at in vivo muscle editing in large animals have been limited by the need for dual vector systems or alternative delivery approaches, highlighting the unique therapeutic potential of NanoCas's compact size. Discussion. Here we present NanoCas, an ultra-compact CRISPR nuclease that challenges a fundamental assumption in genome editing, that smaller systems must sacrifice performance for size. Discovering this system required extensive screening of naturally occurring CRISPR variants, evaluating over 150 candidates in the lab, followed by targeted protein engineering to enhance editing efficiency. The result is a nuclease that matches the capabilities of conventional systems at less than half their size. The discovery of NanoCas provides insights into how evolution can generate unexpected solutions to complex molecular challenges. While Class II CRISPR effectors were thought to require large protein architectures to support their molecular machinery to the power of 5, diverse microbial environments, including Nanorchia, candidate file irradiation, and viruses to the power of 7 feet 16 feet 17, have driven the development of remarkably compact systems. NanoCas and other compact type V systems' elegant solution of forming a homodimer on a single guide RNA represents an architectural innovation that would have been nearly impossible to achieve through traditional protein engineering approaches like those used to miniaturize Cas9 to the power of 18. Our systematic characterization of 150 related CRISPR systems revealed the tremendous diversity of CRISPR activity in mammalian cells, while previous screens of Cas12a, a type V CRISPR system, found significant activity in human cells to the power of 20. We observed substantially lower activity rates among ultracompact systems. The reasons for this difference remain unclear but may involve variations in molecular architecture or cellular interactions. Nevertheless, NanoCas demonstrates that carefully selected compact systems can achieve robust editing across various contexts, challenging the assumption that small CRISPR systems are inherently less effective. We have demonstrated NanoCas's therapeutic potential through both in vitro studies and in vivo applications in disease-relevant contexts. In the liver, we achieved efficient PCSK9 knockdown in mice. More significantly, we successfully edited the DMD gene in both mouse and non-human primate muscle tissue using a single AAV vector, presenting a potential therapeutic approach for DMD patients who currently have limited treatment options. The robust indel formation at splice junctions in these animal models is particularly promising, and NanoCas's success in large animal studies is notable given that few CRISPR systems have demonstrated such efficacy. NanoCas's compact size opens several promising avenues for therapeutic development. Perhaps most exciting is its potential compatibility with non-double-strand break applications like base editing, reverse transcriptase editing, and epigenetic modification. Even these larger editing modalities can be packaged into a single AAV when built on the NanoCas scaffold Fig. 4F. Furthermore, NanoCas's small size, requiring only 2.4 kilobytes including regulatory elements, enables the use of self-complementary AAV vectors, which have shown improvements in transduction efficiency to the power of 21 feet 22. This could potentially allow for lower dosing requirements, improving the therapeutic window and reducing manufacturing challenges. Beyond AAV delivery, NanoCas offers advantages for other therapeutic modalities. Its compact size means shorter mRNA constructs for non-viral delivery, potentially improving manufacturing yields and enabling more efficient packaging in lipid nanoparticles, LNPs. 23. As new delivery technologies emerge, many with their own size constraints. 32. NanoCas's compact architecture may prove increasingly valuable. This versatility could help extend gene editing therapies to diseases and tissues previously considered inaccessible to conventional CRISPR systems. The rapid evolution of CRISPR technology from basic research tool to approved therapy in just a decade demonstrates the field's extraordinary potential. By expanding the capabilities of genome editing while simplifying delivery, systems like NanoCas represent a crucial step toward realizing this potential across a broader range of genetic diseases.
It's an extreme pleasure today to welcome Professor Ian Akyildiz to NYU Abu Dhabi. Professor Akyildiz got his education in Erlangen, Germany, but then most of his career is spent in Georgia Tech, where he has been tenured professor for over 35 years. He is one of the world's most known experts in wireless communication and has been very active around the world and held in position in different countries, including Spain, Finland, Saudi Arabia, and recently Ireland. He is also advisor at Technology Innovation Institute in Abu Dhabi, and that's why we have a pleasure to host him very often in the UAE.
The question here is, can we create man-made man meaning, human-made biological nanomachines? The answer is yes. That means we can reprogram these artificial cells and via DNA manipulations, and that's called genetic engineering. And that will really help us a lot. In fact, unfortunately, some maniacs in the US found all my work the last 18 years. I'm getting a lot of hatred mail, so my life is in danger. They think that I'm Dr. No in James Bond movies, that I'm trying to manipulate human lives, but it's, I'm not the only one. There are many people working on this subject, So now what we try to do is, so you have seen the bacteria, right? So what we can do is we can try to genetically engineer this bacteria, and there are many illnesses, as you know. I will point them out to you, based on bacteria or viruses, right? So if I create genetically engineered healthy bacteria, for example, Crohn's disease, I don't know if you know that, it's an awful disease for intestines. So there's no cure. So I can inject into the body, and then they can fight these ill cells or ill bacteria and then solve the problem, So just an example. So I hope you are getting the point, right? So we are trying to create these nanoscale machines, genetically engineer them. They will act like bacteria, and also in the future we will see that neurons and calcium ions and etc. And it will help us for prolonging our lives. In fact, the mRNA, the COVID injection is exactly based on this idea. So all these fluids, right, there are nanoscale machines inside, right? So when you look inside.
In terms of Bio-Nano things, these are for the health applications. I did also a lot of research on that the last 15 years. Bio-Nano scale machines, but these are for the, injecting into the body and always monitoring the health problems. And that is also really going really well, like with these COVID vaccines, it's going that direction. These mRNAs are nothing than small scale, nanoscale machines, right? They're programmed and they're injected. And then into other nanoscale things. So those will be part of 7G and beyond, right?
Nanotechnology is now pervasive in comprehension of nature, solutions for emerging technologies, and in all economic sectors dealing with matter, energy, and life systems.
One of the main outcomes of the NNI after 20 years of federal investment is one of the key technologies that was needed for these mRNA vaccines. So if you got a Pfizer vaccine or a Moderna vaccine, the only reason that worked is a nanotechnology called lipid nanoparticles.
Not growing the virus at all, but just getting the sequences, getting the appropriate protein and sticking it on a self-assembling nanoparticle that is much, much more immunogenic.
Assessing the brain, not crudely, but ever more creatively. Utilizing other forms of technology and engineering, such as nanoparticulate matter that we can then create into sensors and transmitters that can be inhaled, ingested, rubbed on, that can then be migrated into the brain to form vast arrays of sensors and transmitters to be able to read information from the living brain and write information into the living brain in real time remotely. This is a current project of the Defense Advanced Research Project Agency here in the United States called NQ'd, Next Generation Non-Invasive Neuromodulation. Now that we can transmit into the living brain remotely through the cloud. If in fact we're using the cloud that way, now we're harnessing, if you will, the cybersphere to be able to access the cerebrosphere of individuals, of groups, of collectives. If in fact that same technology that allows us to literally read the mind by scanning and interpreting the data we get from the structures and functions of the brain, and the inverse of that is that we can then write into the living brain, that's mind control. And what will we do with that capability? Where will those data go? What will they be used for? By whom? In what ways?
When the infotech revolution merges with the biotech revolution, what you get is the ability to hack human beings. And maybe the most important invention for the merger of infotech and biotech is the biometric sensor that translates biochemical processes in the body and the brain into electronic signals that a computer can store and analyze. And once you have enough such biometric information and enough computing power, you can create algorithms that know me better than I know myself. And humans really don't know themselves very well. This is why algorithms have a real chance of getting to know ourselves better. We don't really know ourselves. Directly connect our brains to the computer network. But this is wrong. Simply by gaining mastery of human language, AI has all it needs in order to cocoon us in a matrix-like world of illusions. Contrary to what some conspiracy theories assume, you don't really need to implant chips in people's brains in order to control them or to manipulate them. For thousands of years, prophets and poets and politicians have used language and storytelling in order to manipulate and to control people and to reshape society. Now AI is likely to be able to do it. And once it can do that, it doesn't need to send killer robots to shoot us. It can get humans to pull the trigger if it really needs to.
So you can just imagine that in many parts of the world, humans will be hunted. They will be cowering underground in shelters, devising techniques so they don't get detected. They could be here in a few years. This is the easy scenario. There are worse. In the Internet of everything, everything can be penetrated. Everything can be sabotaged. Everything can be subverted. And when I say everything, I mean everything. It's our personal privacy. It's the robustness of our infrastructure. It's our national economies and our national defense. Everything from our personal accounts and information, our bank accounts, our power grids, our communication centers, our planes, our cars, that'll change too in a big way. Even the way we do elections. Everything can be penetrated. This is a fact. In the meaty ground between big data, connectivity, and artificial intelligence, we have vast opportunities. And we have huge industries springing up from, as if from nowhere, almost instantaneously. The meeting ground between big data, IT, and connectivity. Everything is being driven from this nexus. Everything, everything. We stand, all of us, at the nexus of big data, connectivity, and artificial intelligence. Great opportunities, but great challenges. In Israel, we're exploiting these opportunities and we're meeting these challenges. What this revolution is doing, big data, AI, and connectivity, is allowing small countries to be big countries. It depends if you have the brains and the system to create conceptual products at an accelerated rate, conceptual products that are at the cutting edge and give you a competitive advantage. If you do this conceptual mousetrap, the world definitely beats a path to your door. There's no question about it. It's happening. The intersection of big data, connectivity, and artificial intelligence, and what it does in robotics and genetics and all the other fields, Uli, that you mentioned, everything. That is changing our world in rapid, rapid succession. And the future belongs to those who can seize this change. We are positioned right at the cusp of this change, right at the center of this change. We can take it, and we are. It's changing us. It's giving us powers and prowess that we never had before. It is a force multiplier. Because to survive and thrive, we have to overcome a simple problem. The problem is, there aren't that many Jews in the world. Our numbers are small. And history favors large nations, not small ones. So we have to compensate for our relative numbers. And we've always compensated with our brains and with our guts, but we're in a position now to compensate beyond anything that we dreamed of. And that depends on one thing. We think there is a tremendous business opportunity in the never-ending quest for security. It creates its own demand. It grows, probably grows exponentially.
I find it fascinating, and it's actually the new world, and it's also built on the platform concept. You create a platform where everybody can share goods, services, and this will be very much the future. The future will not necessarily anymore a world where people own things, but a world where people share things. Now this has quite some implications on the economy, because you have to produce less goods in order to achieve the same satisfaction. You can share the goods. The World Economic Forum is engaged into a big program to look at the implications. For example, how does it affect productivity increase? How does it affect the measurement of GDP? So it's a new world. It's a fascinating world, and it will be the world of the future. We will share much more what we are now used to own.
We have, if I look at our stakeholders, we have business, of course, as a very important audience, and we have politics. We have continuous partnerships with many governments around the world. And of course, we have NGOs, we have trade unions, we have all those different parts. Media, of course. Media, of course, and very important experts and scientists and academia. Because if we are looking at the future, I think we should look at new solutions, and the new solutions will be very much driven by technological developments. And you even have religious leaders, right? Religious leaders, we have social entrepreneurs, very important social entrepreneurs.
And now, the fourth industrial revolution is not just a prolongation of this digitalization, it's much more. It's a combination of technologies. It's not just the digital technology. Just think of genetics, think of brain research, and so on. And the power of the fourth industrial revolution comes from the combination of all those technologies. Actually, I was saying it's at the end what the fourth industrial revolution will lead to is a fusion of our physical, our digital, and our biological identities. So nobody will be safe if not everybody is vaccinated.
We want to make sure that people can discern the truth from the misinformation. And we want to make sure that everyone understands that no one's safe till everyone's safe.
No one is safe.
No one is safe.
No one is safe. No one is safe.
No one is safe.
Nobody's safe.
No one is safe.
Nobody is safe.
This is a post 9/11 axiom. Safer but not yet safe.
No one is safe. No one is safe. No one is safe. No one is safe from COVID-19 until everyone is safe.
If the whole world isn't safe, none of us are safe.
No one is safe.
No one is safe.
Nobody is safe until we're all safe.
Health experts have been saying nobody is safe.
Nobody is safe until everybody is safe.
Nobody is safe.
The science is clear.
None of us are safe.
There is no safety.
No one is safe.
No one is safe. No one is safe. No one is safe until everyone is safe.
No one's safe.
Nobody is safe.
Nobody's safe.
No one's safe.
We'll never be safe
until we're all safe.
We are never going to be safe.
99.5% of people are safe and will survive COVID-19.
The only positive thing out of this is we should be able to manufacture a lot of vaccines, and
nobody will be safe if not everybody is vaccinated.
you don't have a choice.
As long
long as not everybody is vaccinated, nobody will be safe.
Normalcy only returns when we've largely vaccinated the entire global population. So
Get the fucking vaccine!
You need to get vaccinated, and if you don't, you are going to die.
I know you're vaccinated, you're the smart ones, but you know there's people out there who aren't listening to God and what God wants. You know who they are.
The unvaccinated people.
My message to unvaccinated Americans is this.
If you are the unvaccinated, you are the problem.
You're the problem.
Condemn them, shame them, blame them.
The only people that you can blame are the unvaccinated.
Frankly, we can't trust the unvaccinated.
They should not be part of polite society.
Lunatics who won't take COVID vaccines walking around lawfully unvaccinated? That's psychotic.
If you're willing to walk among us unvaccinated, you are an enemy.
You have no right not to be vaccinated.
You don't have the right to contaminate someone else.
You can't go around pointing a gun in somebody's face, which is what it is when people are unvaccinated.
They are all idiots and losers.
This is a real movement in this country against the unvaccinated.
Jennifer Aniston is cutting non-vaccinated people out of her life.
The unrepentant, unvaccinated.
They should be removed from the hospital.
Those who refuse to be vaccinated should be denied health care.
Vaccinated person having a heart attack? Yes, come right on in. We'll take care of you. Unvaccinated guy, rest in peace, Wheezy.
Some doctors are saying they'll refuse treatment for people who choose not to get the shot.
Why are hospital and ICU resources going to them?
Morons who will not get the shot.
The unvaccinated clowns.
Idiots who think that they can do their own research.
Don't do any of your own research.
Doing your own research is associated with conspiracy theory circles.
This go-it-alone approach, doing your own research, it can have serious consequences.
You should get prison time for even questioning the vaccines.
Can we all stop saying, I need to do my own research?
That phrase, do your own research, 4 words, 4 little words that are hurting America.
Doing your own research hurts America!
Everybody has a supercomputer in their hand that empowers them to do their own research, and that's the problem.
You must not do your own research.
I need to do my own research.
I don't understand what that means. I'm doing my own research.
You can't do your own research unless you're a scientist. Do your own research.
Maybe you've told yourself you're playing it safe, you just want to wait and see since this is a new vaccine. No.
Grow the up and get the vaccine.
Unvaccinated people spread the virus.
Get the vaccine, you dopes!
Don't be get yourself vaccinated. That's just all there is to it.
There's no excuse. No excuse for being unvaccinated.
There's no rational and no emotional argument that adds up against getting your damn shot. There just isn't.
But you made a conscious decision not to get the vaccine.
I also have natural immunity. So for me, personally, this vaccine poses a greater risk than a benefit. I'm also not a risk to any of you. Let's look at the science.
So we see that the natural infection is given greater protection or slightly greater protection than vaccination.
This is a vaccine that was created to prevent severity of disease and to prevent hospitalizations. But the vaccine does not prevent you from getting COVID and does not prevent you from transmitting COVID.
Oh my goodness.
Reality.
Well, no,
we have
that's
seen that.
not so. Come on.
No,
you've been at Fox TV
you don't
too long.
have to joy.
I just really don't think that we should allow this kind of misinformation.
Mr. Sandman, get the vaccine so that you don't get COVID-19. We poke ya while we poke ya, so you don't get corona. When we poke ya, well, we know you're gonna be safe from COVID infecting you.
The virus stops with every vaccinated person.
Do it so you stop spreading terrible disease.
And that vaccine, a key goal is to stop the transmission so that you get almost no infection going on whatsoever.
Expectation for vaccines is not to get infected.
A vaccinated person gets exposed to the virus does not infect them.
When the virus gets to you, you stop it.
You're not going to catch it, you're not going to get sick, you're not going to transmit it.
The vaccine is absolutely bulletproof.
Vaccines block you from getting and giving the virus.
100% effective at preventing COVID.
A vaccine prevents you from getting infected.
It is to keep you from getting it and then spreading it.
We have the ability to stop COVID in its tracks.
You're not going to get COVID if you have these vaccinations.
Biden tests positive for COVID-19.
When people are vaccinated, they can feel safe that they are not going to get infected.
Dr. Fauci tests positive for COVID-19.
Vaccinated people do not carry the virus, don't get sick.
CDC director tests positive for COVID-19.
Get your vaccine.
Pfizer CEO tests positive for COVID. Pfizer CEO tests positive for COVID again. CDC director tests positive for COVID again. Fauci tests positive for COVID again. Joe Biden didn't test positive for COVID again.
Cases up 258%. The majority of these cases are among the vaccinated.
And this area is the most vaccinated part of Massachusetts.
So many fully vaccinated people are testing positive.
So people here are a bit angry.
We are all really concerned about what this means long term.
I was vaccinated and I got COVID, and almost everyone we know with vaccinations had at least one bout of it.
Our vaccines work better than we could have possibly hoped they would work.
We are not the problem. The problem are the unvaccinated.
The unvaccinated threat.
Unvaccinated people are a threat, a direct threat.
The threat to all of us.
The unvaccinated are a risk to all of us.
People have a right to be protected from the unvaccinated.
Maybe there should be laws that allow them to be kept out of the building.
So at least, thankfully, they're not breathing the same air.
Their freedom to breathe will diminish. Start firing the unvaccinated.
If you don't vaccinate, you'll be fired.
Time to come down on the unvaccinated.
They should be banned from the VA, banned from restaurants, banned from other businesses and colleges.
Companies should not treat us as equals.
Place unvaccinated people under arrest.
Who's the unvaccinated that has put America in the place that it is?
We are losing freedom because people are unvaccinated.
Anger towards the unvaccinated. I'm furious at the unvaccinated.
Frustrated with Americans still not vaccinated.
Not to be vaccinated seems criminal.
Suspects face years behind bars for coughing on police officers.
Their freedom.
For them charged with terrorist threats.
You have no individual rights when it comes to the vaccine.
Really, you're killing other people.
We're being attacked by unvaccinated.
The unvaccinated who aren't wearing masks. It's the unvaccinated who aren't social distancing. It's the unvaccinated going to crowded indoor events.
It is not your right as an American citizen to catch and transmit a potentially fatal infection.
So screw your freedom. We can coerce you.
You're not going to be able to travel to see your family. If someone in your family isn't vaccinated, should you ask them not to show up?
Yes, you really shouldn't have anyone unvaccinated come to dinner. That's just a huge risk.
If you want to come out into public, if you want to live your life, you need to get the vaccination.
It's time for people to see vaccination as literally necessary. You should have to show that you're vaccinated in order to go places.
Unvaccinated tennis player can't travel to the U.S.
If you're not vaccinated, you're not welcome.
Our patience is wearing thin.
Why hasn't the president focused more on scolding the unvaccinated?
The refusal has cost all of us.
Continued damage the unvaccinated are doing to themselves and the country.
How come migrants are allowed to come into this country unvaccinated, but world-class tennis players are not?
You know,
And back to our lead story, the
Pandemic of the unvaccinated. Fact is, this is a
pandemic of the unvaccinated.
Experts call it
Pandemic of the unvaccinated.
Still a pandemic of the unvaccinated.
The disease of the unvaccinated.
It is the unvaccinated who are the problem. Period. End of story.
The unvaccinated also put our economy at risk.
This is a tyranny of the unvaccinated right now.
The unvaccinated, not the vaccinated, the unvaccinated. That's the problem. That's the pandemic of the unvaccinated.
Pandemic of the unvaccinated. All this is a pandemic of the unvaccinated.
The only pandemic we have is among the unvaccinated. Pandemic because of the unvaccinated.
When you get the vaccine, you will not die. That's right. That's right.
It's a simple, basic proposition. If you're vaccinated, you're not going to die.
If you're vaccinated, you don't have a risk. It's as simple as black and white.
You are not going to die if you are vaccinated. That's it, full stop.
You're unvaccinated, you're at risk. You're vaccinated, you're safe.
A majority of Americans dying from coronavirus are vaccinated. COVID-19 isn't a pandemic of the unvaccinated anymore.
We didn't really understand the fatality rate. We didn't understand that it's a fairly low fatality rate and that it's a disease mainly of the elderly, kind of like flu is, although a bit different.
We have two Americas, an unvaccinated, at-risk America and a vaccinated America.
I wear my vaccinated necklace all the time to say I'm vaccinated. You, the vaccinated, are the last best hope to overcome the unvaccinated minority. I need you to be my apostles. I need you to go out and talk about it. We are seeking to enlist you in a benevolent conspiracy to join in the unfinished war against the sins of the unvaccinated.
And it's a war.
A forever war. The war against COVID-19.
The vaccine hesitancy on Earth 2 has the potential to compromise all of the hard-fought progress that we've made here on Earth 1.
This is the greatest threat to life that we have ever faced.
Metropolitan areas are now banning all private gatherings. This bar owner arrested for reopening illegally.
Unvaccinated, we are looking at a winter of severe illness and death.
Yeah. For yourselves, your families.
If the unvaccinated are not to blame, Who is?
If only we had a vaccine against BS.
A vital part of preparing for hurricane season is to get vaccinated now. Everything is more complicated if you're not vaccinated in a hurricane.
Is going to get worse.
empty as hell.
No one's in there, guys.
It's absolutely dead. Il y a personne. Regardez, on peut aller fouiller dans les couloirs, on peut faire absolument ce qu'on veut. Je comprends pas.
Not many people were in there at all.
Like a ghost town in this hospital.
We've just been locked down in Gloucestershire for an empty hospital. Look, I am so angry.
So there's nobody even in there? Nobody's in there. Nobody's in the hospital? How is that possible in a medical pandemic?
That's fake news, that's why. That's fake news, that's why. That's fake news, that's why.